and I've been teaching this course for an awfully long time. The average went down by about 6 points, I guess, something like that. And the distribution went from, you had a decent number up here in the first test to lots and lots of people down here. So about half-ish of this class got a D or an F on this exam. Not so super. The other thing that's not so super is that the drop deadline, the late drop deadline is the 15th, which is before our next exam. So short of another quiz and another lab report, which probably is not going to shift your grades substantially, have all of the data that you're going to have to make that decision about whether you're going to stay in the course past the 15th. I can certainly sit and chat with you about it, but the numbers are going to be the numbers, and it all boils down to whether you think you can change this situation, okay? Especially if you're down in this side of things. Some of you need to start thinking about that, considering that decision. Like I said, I'm certainly available to talk to and consult on the numbers, but like I said, the numbers are the numbers, such as they are. Okay, so I'll have these to get back physically to you in lab, and I'll have copies of the question sheets so you can see what you got right and what you got wrong. What else? Live report due this week for vacation. I think it's due this Thursday, and we're doing the GFP lab. So please read that procedure before you get there on Thursday, because it's going to be timed down to close to the second in terms of getting that procedure done in the 2 hours that we have. So I really, really need you to come prepared, ready to go, really pretty much at moment number one. So I also need you there on time, okay? Because otherwise we're just not going to get that procedure done in 2 hours. Okay, so let's stand on that. Finish up translation. So we've gone from DNA to RNA transcription, and we started going from RNA to protein translation. We've looked at the general structure of transfer RNAs that are going to bring amino acids that they have attached at their 3' end to the correct location in the growing peptide. So as we'll see today, we build protein one amino acid at a time, as we'll see also, binding of the anti-codon on the transfer RNA to the codon in the message. So how all that works this morning. The right RNA, transfer RNA and the right amino acid get together through a process called charging. We saw there's a whole sort of family of enzymes called aminoacyl tRNA synthetases that make sure the right amino acid gets attached to the right transfer RNA. And that costs you an ATP every time that happens correctly. That's just the reaction there. We have briefly at ribosome structure. Major ideas here. They are made in two pieces, a large piece, a large subunit, and a small subunit. And they are collections of ribosomal proteins and pieces of a third major type of RNA called ribosomal RNA. And this just compares prokaryotic with eukaryotic ribosomes. They both do exactly the same thing, just eukaryotic ones are bigger than their corresponding prokaryotic counterparts. And ribosomal RNA has usually extensive secondary structure to it. And this is the last slide we looked at last time. When you create a complete ribosome where a large and a small subunit comes together, and you have essentially 4 major binding sites, 1 for the messenger RNA, and then 3 for various transfer RNAs that come in, the E, the P, and the A site from left to right, as this is usually drawn. I think that's as far as we got last time. So, how are we feeling about transfer RNAs, charging them, and ribosomes? We'll put all this together. We'll take silence for you feel wonderful about all this stuff. Okay, one last thing before we can bring all this together is we sort of need to complicate our picture of that messenger RNA we made last time just a little to look more closely at some specific pieces that we didn't really mention before today because we didn't really have to until we get to translation. So we saw transcription, we saw if it's a eukaryotic situation, we have all that processing, right? We remove the introns, put a GCAP, and put a poly A tail. So that is all the stuff we know. Just a couple little pieces that we need to throw in here, and it's vital that we do it because otherwise we can't see how all this is going to come together, is are pieces of this messenger RNA that don't code for protein but are going to do things like align the message correctly on the ribosome, for example. So if you look at it from the 5' to 3' section, there is an initial section on RNAs, whether it's prokaryotic or eukaryotic, it doesn't matter. That's usually referred to as the 5' UTR, the 5' untranslated region. And as we'll see, especially the 5' part, this is going to align the message correctly on the ribosome. So what they're calling here this ribosome binding site is kind of what I'm referring to here. But there is a comparable section on eukaryotic ribonucleic messages as well. So it's upstream of the start codon. So this middle section here would actually be the coding section. Okay, so we would have our AUG here to start and our stop codon down here. So from there to there is the information for the protein. But then there is usually a 3' untranslated region as well. And this can have regulatory function as well. We're not going to pay as much attention to that part. Poly A tail if we were looking at a eukaryotic message, and we know that the poly A tail array extends the life of the message in the cytoplasm. So, that's kind of all we're going to talk about there, but we'll kind of focus a little bit more on the 5' UTR because that's what's going to align it on the ribosome. Does that kind of make sense? And so it's before the actual coding, before the actual start codon. But it's going to be because of this section aligning correctly that the ribosome knows that that is the start codon. That's the AUG, that is codon number 1. Because otherwise, remember, AUG also codes for methionine, so there is presumably going to be other AUGs, so how does it know which one is the first one? It's in large part because of this 5' untranslated region. Because, as we've seen before, this idea of a reading frame, right, because we read it in triplets, we need to make sure that that first AUG is in the right place on the ribosome so that where protein information starts, and it's in the correct reading frame. Because if it misaligns here and it starts here, it starts at the second letter, well then that, in this example here, that's a stop codon, so you're not going to get a protein at all. It starts at the third letter, and you're going to make a protein, but it's going to be a very, very different protein than what it's supposed to be. And you don't have a stop codon in this frame. And so there's lots and lots of good reasons that all of this has to work perfectly so that that first AUG is in its right location so that the whole message is going to be read in that correct reading frame. Does that make sense? And so that's all back to that triplet nature of the code. Okay, so this is kind of the overview picture. We're going to divide it into the same 3 stages that we did transcription. So we're going to do initiation, elongation, termination, just like we did for transcription. It's just, I think, a useful way to deal with it. Okay, so here's initiation. Again, just to remind you, we are looking at the process again in bacteria, just because it's a little bit simpler than it is in eukaryotic cells. But if you understand what's going on here in bacteria, you understand a large chunk of what's going on in our cells. Okay, so here is how this process gets started. So we start with the small subunit. Okay, so initially, the large and the small subunit are separate. So there are large subunits floating around the cytoplasm. There are separate small subunits floating around. Okay, so initially they're separate. And the first thing that's going to bind is a series of extra proteins. Okay, so it's not just the ribosome, the message, and the transfer RNAs. There are going to be other sort of helper proteins or accessory proteins to get translation to sort of work. So these initial factors are going to be called IFs, for initiation factors. And you can see that there are several that are needed to get this process started. And they're just given numbers. So IF1, IF4, whatever. And I'm not too worried about the specific numbers. I'm just interested in knowing that we need some of them to kind of get the process started. Okay, so there are at least 3 of them that bind to the small subunit with energy, with a GTP. So we're going to see that several times today in translation. For some reason, most of the steps of translation require energy in the form of GTP rather than ATP, which is kind of interesting. But energy-wise, it's the same. If you break a phosphate off of a GTP, you also liberate 7.3 kilocalories per mole, just like you do for an ATP. So it's really the same, except that you do need to realize it is a GTP at a couple of steps rather than an ATP. Okay, so we used ATPs earlier to put amino acid and the transfer RNA together. Where was that? There we go. Okay, so we use traditional ATP here, but for the rest of translation, it's going to be GTP, as we'll see. Okay, so we need that. So several of them bind. One of them, at least one of them, has GTP attached, and then this is part of the picture I don't really like. I think the message binds next. Okay, so the messenger RNA hops on, and because of that ribosome binding site, that 5' UTR, also known, at least in bacteria, as the Shine-Delgarno sequence. So it goes by several names. So 5' UTR, ribosome binding site. Shine-Delgarno sequence, whatever. They were the 2 scientists who discovered it. All referring to the same little stretch that has a complementary sequence in the ribosomal RNA that makes up that small subunit. So there's a binding there. And what that correct binding does is that it puts the first AUG, that start codon, in the center binding site, in that P site. And then what binds is that first transfer RNA, okay, that has methionine attached. So if the codon is AUG, then what's the anticodon on that first transfer RNA? Not T, but RNA. Right. The UAC. If we want to attach something here, we need then UAC. Okay, so the anticodon on that first transfer RNA must be UAC, and to transfer RNAs that have UAC as the anticodon, methionine is attached by the amino acyl tRNA synthetase enzyme. So let's start putting this all together. And as you can see, as some things come on, other things come off. So as the message and the first transfer RNA come in, a couple of the initiation factors start leaving. So we're almost there. And then the last thing that comes on is the large subunit. And as that comes on, the final initiation factor leaves and the energy is used. So you can kind of think about it maybe as, we need the energy to kind of hook all of this together into its sort of final form. So by the end of this kind of complicated starting process, we have the large and the small subunit together. We have the messenger RNA properly aligned with the first codon, the start codon, the AUG in the P site. we have the first transfer RNA in place, and we know AUG codes for the thionine. Okay, so let's freeze all this together. Yes, ma'am.
What did you say leaves?
Well, as you can see, the initiation factors are coming off as other things are coming on. Okay, so there's our sort of final form, and in this form we can now start continuing to make our protein.
One large subunit comes on
Everything else. Number 2, and we use the energy. So just kind of read this from top to bottom. Right? The arrows are coming off, right? So 1, 3 came off there, 2 came off there, ATP is used there. Good. Okay, so now we're actually ready to build the rest of the protein. So we go from initiation to elongation. Don't get too afraid, this isn't as bad as it looks. Okay, so quickly sketch what we have here. Okay, so we have our large and our small subunits combined. We have our message aligned correctly. We have our first codon in the P site. We have our first, our first transfer RNA in, and that has thionine on it. So that's kind of where we are. Those are A sites. And so our P site is filled, but our A site is available. So let's just pretend, since I know what it codes for, let's pretend our next codon is GGG. And I just picked that because I know what it codes for. GGG happens to go for glycine. So we need to get our next transfer RNA with glycine attached into this A site. Right? So we need a transfer RNA that has CCC as its anticodon, and if that comes in, it's going to have glycine attached to it. And the way that that's going to happen is with another protein. So we've had IFs, initiation factors, but we're in elongation now, so they're going to change to become EFs for elongation factors. And the one we need to talk about here is one that's called EF2. Not the number, unfortunately, but this, I guess it's Greek. I guess it's a Greek letter, I'm not sure. TU. And its job is to grab onto random transfer RNAs and bring them to the A site of the ribosome. Because we always want to impose like brains on these things, so somehow it magically knows that GGG is there, and so automatically brings a transfer RNA with CCC. It doesn't work like that. It's much, much more random. So this EF2 is just bringing random transfer RNAs and sticking them in the A site. And the only one that's actually going to stick, in our example here, is one that has what? Has CCC. So all the other 60 transfer RNAs won't stick there. They can be brought there, but they're not going to stick, so they're going to float back out. So this process really is much more random than we might want to admit to. So it's just bringing in random ones. It's doing this really, really fast, by the way. Let's see how fast. Well, I don't know how fast that's happening, but prokaryotes are putting in 15 correct amino acids every second. So if they're doing this randomly, so a lot more transfer RNAs are coming in than are sticking. So this is happening really, really fast. So in any event, this EF2 is doing this, so it brings transfer RNAs randomly into the A site. When the right one is brought in, it sticks, right? So let's pretend that happened. So it finally gets around to bringing in CCC. That sticks, right? So that's perfectly complementary. That has glycine attached. When the right one sticks, you get charged, an energy molecule. So as you see here, it brings in, or it uses a ATP. But you only get charged, luckily, that energy currency when the right one sticks in, which is great, because otherwise, man, it would take so much more energy to make a protein. So you only get charged that GTP when the right one actually fits in. Okay? So let's say the right one got put in. Okay? We get charged that GTP. The EFTU goes back and keeps doing this, so it's recyclable. So now we have both our P site and our A site filled with transfer RNAs with two amino acids there. Now we want to make the bond between the two amino acids. Now I'm going to ask you a question that's going to make you remember something all the way back to probably August or maybe September, probably September. What kind of bond do we make between amino acids? Peptide bond, very good. Okay, so then our next step is to make the peptide bond between our first two amino acids, in our example here, methionine and glycine. That occurs through the work of the large ribosomal subunit. It has an enzymatic activity called peptidyl transferase. And that enzymatic activity that we think is actually shared between some of the proteins and some of the RNA, actually, of the large ribosomal subunit, is going to make that bond. So it's going to create a peptide bond between methionine and glycine. I think that's the next slide, yes. Okay, so here's how that works. Okay, so here's our transfer RNA in the P site. Here's the one next to it in the A site. And here's the reaction. So the amino group of the amino acid in the A site interacts with the carboxyl group of the amino acid in the P site, and that creates our phosphodiester bond, our peptide bond between the two amino acids. Right? There's our CN bond that we all know and love and still are able to draw if I force you to do it. Now notice, though, that both amino acids are now attached to the transfer RNA in the A site. So now the transfer RNA in the P site is now empty. So let's see how we can draw this. So I'm just going to create the line there. So there's the peptide bond. So now both amino acids are attached to the transfer RNA is now empty. And the final part of this cycle, go back a slide. So there we made the bond. Both amino acids are attached to the second transfer RNA. The next and final step of the cycle is another elongation factor called EFG. That's helpful. At, again, the cost of another GTP, is going to translocate, to move the ribosome. Well, it's kind of unclear whether the ribosome moving along the RNA, or is it the RNA being dragged through the ribosome? I don't think we know. It probably doesn't matter. But something's moving relative to the other thing. Right? So we're going to move it over exactly 3 nucleotides, exactly 1 codon. Right? And that's going to cost us energy to do that. So however you want to think about it, doesn't really matter to me, but something's moving relative to the other. So we get this situation. So what was in the P site, exit, right? It's going to exit. What was in the A site moves to the P site. So I'm just going to draw that one if you don't mind. So there is our second transfer RNA. And now let's pretend our next one is CUA, since I know what that goes for too. And we have lysine. And just put the N in there to remind you that we make the protein from the N terminus to the C terminus. Okay, so that first transfer RNA has moved into the exit and exited. What was in the A site is now in the P site. Now we have an empty A site, and we start at the top of the picture again and go through another round. All right, so EFTU is now going to start bringing in transfer RNAs again. If the codon is CUA, what's the anticodon? GAU. So eventually, a transfer RNA with GAU is brought in. You get charged to GTP, and that brings leucine. Peptidyltransferase does its thing. Breaks the bond between the amino acid and the second transfer RNA, makes the peptide bond between the second and the third amino acid. EFG comes on, we get charged another GTP, and we move everything over to the next codon, to the fourth codon. And now we have our 3 amino acids attached to the third transfer RNA, and we just keep doing this apparently 15 times every second in bacteria. So again, this is all happening really, really fast. That's the basic elongation cycle. Bring in a transfer RNA, you make the peptide bond between the 2 adjacent amino acids, you translocate everything down one. What was in the P site goes to the E site and exit. What was in the E site. Okay, so you need to focus on these 2 elongation factors, EFTU and EFG. So, if we start adding up some energy, what's our total cost per amino acid in this protein? Okay, we need 2 GTPs for this. We had another cost earlier, right? Right, this is elongation. We got charged 1 GTP here and 1 GTP here. So that's 2, but we had another cost earlier. What else costs us?
The IF factor and we needed 2 more from the other one, from the initiation.
Well, only 1, and actually that to me is the equivalent to bringing in a sense, that's the equivalent to bringing in that first one. So we can kind of ignore that. But we had another big cost. And you all told me you understood earlier. Right? We get charged an ATP every time we get the amino acid and the transfer RNA together. So there is the equivalent of 3 energy molecules, 1 ATP and 2 GTPs for every amino acid makes up the protein, give or take. Okay, so it's a rough equivalency. So that's a lot of energy, right? So if the protein is 1,000 amino acids long, then to make that protein costs 3,000 roughly energy molecules. Okay? That's a lot of energy. So we can start seeing, perhaps, since proteins are made sort of constantly in your cells, how we can, go through, 10 million ATPs every second. Well, this is a big contributor to that cost, right? The equivalent of 3 ATPs or 3 energy molecules per amino acid. That's a lot. Okay, so we're just going to keep going through this cycle over and over and over again until we hit what? Until we hit a stop codon, right? Because that's going to tell it's going to tell the ribosome, okay, we're done, we made the protein. Okay, so eventually, one of the was eventually going to hit a stop codon, one of our 3 stop codons. The other thing I want to make sure we're pointing out here, remember we make the protein C-terminus, so that the thionine is the N-terminus, and we are, notice the direction we're reading the messenger RNA. So we're reading the message 5' to 3', which is a little bit different, I think, than we're kind of used to. There's that aspect of the genetic code. What do we call that characteristic? 5 to 3, N to C. What's the characteristic called for that part of the genetic code, or that aspect of the genetic code? Oh, my dear friends, please, please, please do not think you're going to wait until Wednesday, November 20th, and somehow pour all this information into your little brain. It's not going to happen. Right? You need to be going over this and over this and over this. Repetition is how we learn stuff. Right? I've had this conversation with a few of you personally already. Let's have a quick little aside here with all of you. Okay? I doubt that anyone's going to sit here and tell me you learned how to ride a bike the first time you got on a bike, or you drove your car perfectly the very first time you got behind the wheel, right? Or you made your first 75 free throws the first time you picked up a basketball, right? That's not how it works. How does it work? What do you got to do for all those things? Practice. Learning this is no different. You want to think it is, you want to treat it like it's really, really different than anything else, but it's not. You're trying to learn something. Learning things by repetition is the best way to learn everything, including cell and molecular biology. I can send you hundreds of links to hundreds of research papers that say that very thing, and I will if you want me to. But I would rather you just believe me on that and spend your time going over this over and over and over again. So, back to my original question. What do we call this characteristic of the genetic code?
Collinearity.
Collinearity. Okay. Have to have those, what are they, 5 or 6 characteristics down cold, including collinearity. Okay, so the ribosome is reading along the message 5' to 3', making the protein from its N-terminus to its C-terminus. There's inherent polarity to essentially all biological molecules, as we've seen already in the course. Okay, so we're just turning along, putting adequate for however many codons there are. And we saw already when we studied protein structure a long time ago, some proteins are very small, 8 or 9 amino acids, some are huge, tens of thousands of amino acid clumps. So it's just going to chug along until it reaches one of those stop codons. Okay? UAG, UAA, or UGA. Now, I think we already mentioned that there are no transfer RNAs for the stop codons. Right? There are only transfer RNAs for the coding codons. And that's actually code for an amino acid. So we need something different then that's going to recognize the stop codon, because it's not going to be a transfer RNA. What it is going to be is a third type of protein. So we've had initiation factors, we've had elongation factors, and here we would love to call them termination factors, right? Because that would be wonderfully consistent, and that would make sense, and that would be helpful. So you know that's not what we're going to call them, right? So that's not what we're going to call them. Now, unfortunately, instead of calling them what we should, termination factors, they are instead called release factors. So instead of TF, they are RF. Sorry about that. And it turns out that there are 2 of these proteins, and they're luckily just given numbers, so there's RF1 And there's RF2. And they're looking for different stop codons. So release factor 1 is looking for the two UA ones, so UAA and UAG. And the release factor 2 is looking for the other one, is looking for UGA. So presumably, they're hopping in, wrong direction. Here we go. So presumably these release factors are coming in all during elongation as well, because again, there's no, this ribosome doesn't have a brain. It can't say, oh, I see the stop codon, 4 codons down, I'm going to send out some signal. It doesn't work that way, right? So presumably, as EFTU is bringing in amino acids, these release factors are jumping in occasionally as well. But again, they're only going to stick if there's an actual stop codon. Okay, so let's pretend we finally got to the stop codon. Okay, one of the 3, whichever one, and the correct release factor comes in. And that binding of this protein into the A site of the ribosome, kind of like Rho factor does for transcription, it kind of tells the machinery, in this case the ribosome, okay, we're done. So it's going to cause the ribosome to break the final bond between the last amino acid and the last transfer RNA, because you don't want a transfer RNA dangling off the edge of your protein, right? So we need to break that bond right there. That releases the protein. And then once the protein is released, kind of everything else just sort of disassembles. So the large and the small subunit detach from each other. That releases the RNA. That final transfer RNA is released, and the release factor pops off as well. And as you can see, stopping the process costs you a GTP as well. So again, we still maintain overall roughly that 3 energy molecule per amino acid cost, generally speaking. And then it could all start over again. So assuming this messenger RNA is still good, meaning that we haven't chopped off enough of those A's or whatever, if it's still good, it can start all over again. So the same message would start all over again. There we go. And the cell makes a second copy of the protein. And it presumably does. Okay, so it's actually quite common for you to get multiple copies of the protein off of the same messenger RNA. So these are reusable to an extent. Okay? But we know that eventually, because of those nucleases, eventually enough of that message is going to get eaten away that you can't make the protein anymore. But that usually takes enough time that you can, again, get a few copies at least of the protein made off of that one template, that one message. So that's sort of the short story, at least, of how we get from RNA to protein in translation. So we sort of decode the message in that messenger RNA into a polymer of amino acids, which is the definition of protein. And now this peptide, now let's link this back to the stuff we did back in September, right? So this peptide will start folding right into secondary structure and tertiary structure at least. If this is a part of hemoglobin or part of an antibody, then it's going to hook up with other proteins in quaternary structure to make that final actual protein. So link all of this back now. We've sort of filled in that gap between how the cell makes the protein and protein structure that we studied earlier in the semester. So let's put some of this into motion.
In translation, a cell reads the message in a messenger RNA molecule, mRNA, and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anticodon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site.
Now, of course, we're doing this much more simply, right? They're not showing us the elongation factors. It looks like magically the right one just came in. But again, remember, the reality is the FTU is bringing in random transfer RNAs. Only when the right one is brought in does it stick. Okay, so this is significantly simpler in terms of the animation than we just studied.
The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated. Three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
So a little bit simplistic, but I think it does the overall job of kind of showing us the major things. This is just sort of the same idea, just an animation I got from a different book. This jumps over initiation, so the initiation is done, basically, so the large and the small subunit are together. The first AUG is in the P site, the E site is kind of over here. The first methionine-bearing transfer RNA is in its site, so we're going to kind of pick up with elongation. But again, it's going to be simplified in that they're not going to show us the extra proteins. Our A site is empty. EFTU is bringing in random transfer RNAs, finally brings the right one in. I like this because it shows you the actual chemistry of the peptide bond making. So there's our peptide bond, and now our first two amino acids are attached to the second transfer RNA in the A site. We get translocation. It was in the P site, exits through the E site. What was in the A site is now in the P site. Our next codon is in the A site. Here's our third transfer RNA. We connect the second and the third amino acid attached to the third transfer RNA, and this just keeps going until it hits a stop codon, which they're not showing. So this is really just elongation. So we build the protein one amino acid at a time, starting at the N terminus. Working our way towards the C terminus, reading 5' to 3' in the message. So all of these animations are up on Angel. Take a look at. Okay, we did that one earlier, so we'll show that again. Okay, so that's kind of a relatively short story of how proteins are actually made in the cell. Before we're going to take a little bit of a step to the side. We're going to pick up with these proteins again. because the next thing we want to consider is how do different proteins get to where they're supposed to go in the cell. Okay, so we're going to invoke the endoplasmic reticulum and the Golgi apparatus, and lysosomes are going to be our next big topic. And I want to take it just a little step to the side before we do that and talk just a little bit about how the cell controls which proteins or which genes are active and inactive in a cell at a given time. So that could be an entire course just on that one topic. We're just going to spend one class period on it, just to give you a quick little sense of one way that the cell has to do that. Okay, so on Wednesday we're going to look at something called an operon, which is how bacterial cells control what genes are on or off. And I think it's a very clever system, and I think it's understandable in one class period. So we'll do that as just one quick example. And then on Friday, we'll pick back up with these proteins and see how they're sorted to their right location. So does that protein stay in the cytoplasm? Does it go into the mitochondria? Does it go into the nucleus? Where does it end up going? How are they sorted? Okay, so that'll be our next big topic. Okay, so you're going to go home and look at all this stuff and remind yourselves about collinearity and other aspects of the genetic code. And we'll continue with this journey on Wednesday. We're working on your clavicle ornamentation.